Thanks, Chrissy, for that very kind introduction. So I know this is probably predominantly a mining audience, so many of you have probably not heard of Argenica, but we think it is a really exciting drug development company that is on the brink of pushing through to clinical trials and starting to treat patients. So just to give you a quick overview of what we're looking to do. So we are developing novel neuroprotective therapeutics to protect the brain following injury. So we're looking at things like brain injury following ischemic stroke, traumatic brain injury, and hypoxic ischemic encephalopathy. And really the driving force behind the company to do this is to positively impact patients' lives. So these conditions are absolutely detrimental on these patients following these injuries. But obviously in doing that, we are untapping a huge commercial opportunity for the company. There are currently no drugs on the market that are able to protect brain cells following injury, such as after stroke and TBI. So just to give you, now that I've given you that snapshot, kind of some of our key company metrics. So we did list in June last year, 20-cent IPO price. We currently have, at 30 June, 8.9 million cash in bank, and that doesn't include the 1.38 mil R&D tax that we recently received. So this funding is enough to get us through to the end of our phase 1 clinical trial and to start preparing for a phase 2 trial in stroke. We're sitting around 41 mil market cap, share price closing at 45.5 cents today. So a really strong share price performance since IPOing in June. So what is our drug? What are we doing? Our lead drug candidate is ARG-007. Now, whilst we only listed in June last year, this has been 10 years in the making by our Chief Scientific Officer, Professor Bruno Maloney. Bruno's based at the Perrin Institute and the University of Western Australia. So he has published 25 peer-reviewed publications on the efficacy, the safety and the mechanism of action of this drug. And that is quite phenomenal for a biotech company at the stage where we're at. So this de-risks our asset through scientific rigour, which we are really proud about as a company. It has allowed us to get ethics approval for our phase 1 clinical trial, which we announced that we have dosed our first patient, and they are still alive. So we are very happy. So I'll talk a bit more about the phase 1 trial in a minute in terms of what that means. The IP is 100% owned by the company, so even though it came out of the Perrin Institute, we do own it, and we have multiple granted patents in our key markets, which really are the US, China, Europe, and Japan. So how does the drug work? So if I talk about stroke specifically, when someone has an ischemic stroke, it's caused by a clot in a blood vessel in the brain. That clot prevents blood flowing to that part of the brain and deprives the brain of oxygen. And that's why you get that cell death in the brain cells at that area of blood flow restriction. But what happens from there is that each brain cell talks to the next brain cell, to the next brain cell. And you get this proliferation out, or this cascade of neuronal cell death. And there's a term, and I think the Envision guys used it in a session previously, that time is brain in stroke. So for every minute that you delay treatment, you get 19 million brain cells dying. So it's really critical to address the problem. But the problem with that is you've got to know what sort of stroke that you're having. So in all this delay, you're getting this cascade out, this increasing proliferation of neuronal cell death. So what we've shown through all our preclinical work is that our drug is able to stop that proliferation of cell death in the brain and buy that patient time to get the treatment that they need. So as I said, we've got extensive preclinical data, and one of the most striking papers is in non-human primates. So this is in a stroke model where we've showed when we deliver our drug 2 hours after stroke, we're getting a 66% reduction in that brain tissue death. And that's compared to our nearest competitor, NA1, who are currently completing phase 3 clinical trials. But what's probably more striking is that effect, even though it's only a single injection, it's lasting out to 28 days post-stroke, which is really important in terms of the functional recovery of these patients. So as a company, why are we focused on stroke? So 1 in 4 people in this room will have a stroke in their lifetime. That's pretty significant. It's 15 million people a year. And of those people that have an ischemic stroke, only around 10% will recover fully. And there are no drugs on the market that can protect your brain following a stroke. So we are hoping to become a leader in this area of neuroprotective therapeutics. So just to give you an understanding of the clinical workflow when someone has a stroke. So if you have a stroke and you call an ambulance or your loved one calls an ambulance, there's very little that the paramedic can do in terms of triage. So the aim of the game is to get that person to hospital as soon as possible. And the reason for that is because you have to diagnose the type of stroke before you administer treatment. The 2 types of treatment for ischemic stroke are a clot-dissolving drug or mechanical thrombectomy. So a neurointerventional radiologist going in and mechanically removing the clot from your brain. But if you give the clot-dissolving drug to someone that's had a hemorrhagic stroke, so they're having a bleed on the brain, it can exacerbate the bleeding and it can be fatal. So hence the need to diagnose. But if you think about someone that has a stroke in Broome, they need to get to a stroke care unit, which is at Charlie's or Fiona Stanley in Perth. That takes time. You get to ED, you call the Royal Flying Doctors, you come down to Perth, you get wheeled into ED there, you get diagnosed. All the while you get these increasing levels of cell death. So what we're hoping to do is to have our drug administered by first responders, by the paramedics, by the emergency doctors and nurses, to buy that patient time to get to the stroke care unit to get the treatment they need. So as I said, really encouraging results to date. We've got over 13 publications in stroke showing the efficacy of our drug in various animal models. We know that it's safe to administer in the field, so we have published data to show that it doesn't exacerbate bleeding in hemorrhagic stroke. So we can give it to a patient regardless of the type of stroke, so before diagnosis. And we've done extensive preclinical work to show there's no adverse effects of our drug in animals at the efficacious doses. And one critical point when anyone's developing a drug is it has to be able to fit into current clinical workflow. So we know that our drug is safe to administer with the clot-dissolving drug that patient may get later on. I do say that because our nearest competitor I showed before, their drug can't do that. So it is a key competitive advantage of ours. So just in terms of the phase 1 objectives, so the phase 1 trial is in healthy volunteers. We are still looking for more healthy volunteers, if anyone feels free to put their hand up. It is really to test the safety of our drug. Is it safe in humans? How does the drug work in the body? And what is that ideal safe dose that we take into a phase 2 clinical trial? We've put out quite a number of announcements on the design of that phase 1 trial, but just quickly, it's pretty quick. So we've got 4 cohorts. Each cohort is blinded, so we don't know if they receive our drug or they receive a placebo. The first cohort that has now been dosed, they receive the lowest dose of the drug. They stay in at linear clinical research in Perth for 48 hours. They come back on day 8 and they get more bloods taken. We get approval to move to the next cohort, the next highest dose, and so on. So time frame for completion, we will have our final dosing of our last cohort done by the end of this year, early next year, which gives us then the data that we need to put in an ethics application to go into our phase 2 clinical trial, which will be in stroke patients. But importantly, the mechanism of action of our drug lends itself to a whole raft of different neurological conditions. So the ones that we're really focusing on are listed here, but where we've got quite compelling data is in hypoxic ischemic encephalopathy and traumatic brain injury. So if I talk about traumatic brain injury, this data that you see here is in a more severe traumatic brain injury model. So that would be equivalent to domestic violence, road trauma, that sort of thing. And we do have a whole program of work coming up around sort of mild to severe traumatic brain injury, where mild TBI is really related to concussion. But we're really excited about the HIE data that we have. If I just quickly go to the next slide. So we recently announced this data. So HIE is what's termed a rare paediatric disease. As I said before, it affects infants, newborn infants. So it's usually as a result of things like preeclampsia, the cord around the baby's neck during labour. But the outcomes for a baby that sustains HIE can be absolutely awful. So severe cerebral palsy, severe learning difficulties, severe motor dysfunction. And for these babies, at the moment, the only standard of care is hypothermia. So to induce hypothermia, to cool their bodies down, and which is an amazing neuroprotectant. So it does stop that cascade of cell death, but it also shuts down all the other organs in the body. And in this case, it's not particularly effective. Now, it's a rare disease, which means that under FDA, there's less than 200,000 babies that have this condition. But from a commercial point of view, it gives us an accelerated clinical development pathway. So the FDA has said, if you're going to research and develop a drug in a rare disease, we want to accelerate that approval pathway, and we'll also give you a rare disease paediatric voucher, which means you can sell onto other companies to get accelerated pathway for any condition. So this data is really compelling. In this animal model of HIE, at our 300 nanomole per kilogram dose, we showed an 86% reduction in that brain cell death, which can mean the difference between the ability for a child to walk or be wheelchair bound. So we're really keen to progress this into the clinic. We have a few more preclinical studies we need to do for this indication, but we can use that phase 1 adult data to move straight into a phase 2 in HIE. So just on to the team. So really fortunate to have, thank you, an experienced board and management team. So with a great depth of experience around not only capital markets, but drug development and taking drugs all the way through the drug development process. And for our stroke, we're supported by global world-leading clinicians. We recently announced the appointment of Dr Jeffrey Saver, who is based at UCLA. He is the stroke trial guru. He has done over 50 clinical trials in stroke. And when we actually approached him, he was so excited. He's been following Bruno's work for a number of years. And the leading guy is Professor Bruno Maloney. He's the inventor of the drug. He still does all the research for us at Perrin. And he is an amazing, he's been working on this for 20 years in the area of stroke, and he is an absolute superstar. So we're so delighted he's still involved in the company. So just to wrap up, key investment highlights. We feel that we have de-risked our asset through rigorous scientific validation, which is all peer review published. We have a really strong balance sheet to fund to the end of the phase 1 clinical trial. We are not a one-trick pony. We have multiple applications of the drug in some huge markets and some really exciting near-term catalysts supported by a really experienced board and management team. So thank you. And come and see us. We're in the hot box down the bottom, so feel free to come and join us down there.